Okay, everyone, I can see that we have still a lot of attendees at the workshop. We are going now into the more exciting part of the workshop where we will try some hands-on coding and some live coding and some live examples from Jonas' side. Jonas, he has been part of the testing panel when we have put up AlphaFold version 2 and RoseTTAFold on the University of Oslo machines. We will also have Sabri here, who can also answer technical, if you have any questions about the technical aspects of this. Sabri will also talk about this tomorrow, parts of how to install and what kind of challenges he has met along that path. Jonas is now logged in as to Jonas, I can see, so that I guess means that you are soon ready. I should also note tomorrow we have very many nice sessions. Brandy Reed, who was having the previous talk, will also be in a panel debate. Min Kyung Baek from the Baker Group, who was central in the RoseTTAFold paper and work, will have a talk, which I'm really looking forward to. We will have many great talks tomorrow. But now it's Jonas. Jonas, he is a doctoral research fellow at the University of Oslo. He's a computational neuroscientist who focuses on several novel methods to study and cure psychiatric disease through the application of a wide array of computational techniques at the University of Oslo. As part of the 4-month life science convergence environment, he studies protein structures and levels derived from RNA signatures in brain organoids. Before taking up this position, he worked in the pharmaceutical industry and as a research assistant in theoretical physics, where he developed computational methods to study highly entangled system. So Jonas, I'm really looking forward to this, and this will be exciting to see how far we can get in the live demo session.
Okay, thank you. Just trying to see if I can hear me. I'm trying to see if I can share my screen. There seems to be a final technical issue. But,
yeah, Jonas, I think from our side the settings are fine. But are you getting any trouble sharing?
Yes, it's not allowing me apparently.
Okay.
I'll try again. Okay. I'll start by sharing on this.
I will suggest while you're solving those issues, if it's Sabri, could you also turn on your camera?
Yeah.
Tomorrow, Sabri, yeah, it's all solved now. But tomorrow, Sabri.
No, it's not solved, but okay. I can show the presentation at least for now, and this I can do from a local machine. Yes, class, you were saying.
Yes, no, I was just thinking maybe Sabri could say something about kind of the access. He mentioned it in the break, but unfortunately we have had so many people attending, or fortunately we have had so many people attending the workshop and wanted to sign up for this live hands-on coding. So we have unfortunately not been able to allow everyone, Sabri, but it would be possible also for more people, if I understood correctly, to get access tonight or tomorrow. So maybe you could say just 2 words about that, Sabri.
So the current system, the machine learning nodes that we're going to demonstrate, that is inside our university network. So it's only for the University of Oslo researchers and students and collaborators. So we cannot grant access to outsiders, but what we could do is we could try to reproduce the setup on some sort of cloud where we can give access to outsiders. So that we are not fully thought through yet. So what recommended is to follow the procedure, or they could use one of the national systems like Saga if they're already in Norway, or they could follow the same procedures in their like local systems. So I can't promise that we could arrange access to outside participants inside this infrastructure. So I had to apologize on that. So it's not about like a policy. It's a technical issue that you need a University of Oslo account in order to access these things.
Okay.
But but for the mailing list, we'll come back with some sort of solution. So at the moment, you have to just follow the instructor on screen.
Excellent. We will go on following the instructions here then, and we will see also if the issue with people from outside Norway can be solved further down the line. So Jonas, please.
Okay. So I'll start off with a very brief presentation, or we'll just discuss kind of what you see when you run the code and what's happening. So yeah, thank you for the invitation. I look forward to doing this. Let's hope that TAC is with us today. So as you've heard this morning in great depth, what we're just trying to do is put in a simple amino acid sequence, get out the structure, the three-dimensional structure. This is important because that gives you an idea about the function and in case of diseases, how to treat them, and in other industrial applications. It might, in the long term, help you get towards de novo design of proteins or, for instance, enzymes with important applications. I'm just going to briefly mention this again, the multiple sequence alignment. I know we've heard a lot about it already today, but it is a very important idea, and it will take up most of the computing time that you spend on predicting these structures. So what is a multiple sequence alignment is you take several sequences of proteins, could be from different organisms, to amino acid sequences, and you compare them basically to the amino acid sequence that you're interested in. So you get all of these out of the database, and you compare them, and you might learn things about what's allowed and what's not allowed in these combinations of amino acid sequences based on what evolution has tried. And you see these two tools I just highlighted, jackhammer and HH-bits. There are a few more. Basically, anytime you see something called hammer or something that has a capital double HH in it, that means that you're looking through some of these databases and trying to form a multiple sequence alignment. And you will see when you run the code and you get some output, that it will spend the majority of its time in these programs. And an interesting thing that especially is coming out of AlphaFold 2, and it's also been mentioned a few times this morning, but I want to stress again, is that out of all the different techniques that exist for finding protein structures, it turns out that just having a good MSA is kind of crucial, at least for both RoseTTAFold and AlphaFold 2. And at anything below 30 or 50 comparisons, so other sequences to check against, makes it very hard to predict a protein structure correctly. And anything above 100 of a few hundred actually doesn't make that much of an improvement anymore. So finding good MSAs, qualitatively diverse MSAs, seems to be a crucial insight. And yeah, there's the drawing in here is, can you hear me? Oh.
We can hear you, Jonas. Yes.
Good. Okay, I'm getting.
But we cannot see you. So if you're able to make you visible as well, that's.
That's not normal.
No.
But we have a few technical issues because I'm running on two machines at the same time, which normally shouldn't be an issue. But I'll sort it out. I'll just briefly finish my explanation first.
Absolutely.
So the only thing that you need to know about MSAs is you're comparing basically what you see in nature of which combinations of amino acids in a sequence can happen, and they give you some kind of constraint on how the three-dimensional protein can be. And that's actually the most important thing to get. And then how protein prediction used to happen back in the early days, before all this deep learning, would be start off from this MSA and then try to figure out kind of, okay, how these residue contacts, these kind of limitations on how your protein can fold, what are they? And then also use this multiple sequence alignment to come up with protein fragments and templates. So protein fragments are small pieces of protein that you kind of know what the three-dimensional structure is of. And templates are larger proteins that you could kind of use as a starting point to try and figure out what your folded protein is. And if you look at the AlphaFold 2 pipeline and later at RoseTTAFold pipeline, you kind of see the same things only more complicated and put into a deep learning pipeline. So you start off with an input sequence, and then you calculate an MSA. As I said, that's the majority of the time of your program is going to be spent on that. And then this MSA representation just gets fed into your deep learning architecture as a whole. At the same time, you're looking at these pairing structures, figuring out how certain pairs, which distances they should have, how to relate to, and you do a structural database search for templates or smaller parts of proteins that you can use to learn from. The deep thing that we've learned from AlphaFold 2 and RoseTTAFold is that it is really these MSAs that are crucial in learning. What you get as an output by the end is a nice three-dimensional protein structure and confidence levels. It's been discussed by previous speakers as well. I'm just showing you this again, because when you're running it, you will see the system go through all these steps. So you will have to search through the MSAs, and then you will see it go through the evoformer, the structural module, and then doing the recycling, where basically you're using the predicted structure with its uncertainties as a kind of template input to the whole system again, and you go through it a few times. An interesting point here is that when you're dealing with very tricky proteins, people have been experimenting by extending this recycling period. So instead of doing it 3 times, doing it 5, 6, 7 or more times. And actually you kind of converge, at least in certain examples, it's been shown that you can actually converge on the right 3-dimensional structure. And the funny thing is, or the interesting thing is that the model knows this. So it's telling you it has a low confidence, and then you run it 3, 4 more times. And by then it actually knows that it's getting it right and it has high confidence in its structure. So I suspect in the future, that will be a hyperparameter that people will learn to auto-tune to some extent. And you will just see the output of AlphaFold 2 going through all of this. And the same with RoseTTAFold when it's going through this step. So it's handy to keep this in mind and you understand what's happening. There's a lot of explanations already about AlphaFold 2, but it is a very, very technical program. A lot of engineering has gone in it, the basic ideas, except maybe for this recycling, and some deep learning ideas have been around before, but it is really a major effort of how to engineer them, how to combine everything. And there's this nice blog post that's been doing the rounds last couple of days from one of the UK groups where they talk about what's new behind the structure prediction, and they actually go into detail of some of these tricks that the engineers have used to make this work, and they explain it in fairly understandable terms, and then point you to the parts of the appendix of the original paper where you can look at. Just to give you one example, this morning they already talked. Somebody already mentioned fact that you're using higher dimensional structures, these quatrions, to optimize your rotations and so on efficiently. But another thing that I've done, for instance, is dealing with large MSAs. It's hard if you want to use an attention mechanism on it. So they separated the rows and the columns, and then the order in which they do it, and then combine the rows and the columns of these big matrices is kind of important. So if you're interested in the technical details for your own projects or just in general, I kind of advise you to have a look at this blog post, and some of the output of the AlphaFold and RoseTTAFold will become more clear. Similar RoseTTAFold pipeline is not that massively different. They're putting in MSAs, they're putting in templates and sequences. They apply attention first in a 1D or 2D block. And then they have this part where they combine the first predicted structure with the information from the MSAs and the templates. And they will mix this together to get an output. So AlphaFold 2 does this through this recycling, and the RoseTTAFold pipeline does this in this so-called 3D track. But we'll have a very nice talk tomorrow explaining this in detail. And just so you know that there are technical details, and it is all, I mean, the devil is in the details in these things. And there are different algorithms, but a lot of the same ideas come back. It's applying attention to MSAs, to some extent to templates, making a first 3-dimensional prediction, and kind of using all of those 3 components together in a rotationally invariant way to come up with a full atom model. Implementations, this is why we're here today in this talk at least. So I'm pretty sure that most of you have seen these notebooks flying around on the internet. There are 2 main ones, the original AlphaFold Colab. This is provided to you on Google Cloud by Google. This is the version that the DeepMind people have set up. It is strongly limited in what it can actually look at in terms of MSAs, in the sense that they only use, I think, the smallest or the second smallest of the MSA databases that they use in the full setup. The reason they do it is because otherwise it would take a long time to actually run these things in a collapse in what's basically a Google hosted Jupyter notebook. As a reaction or in relation to that, there were the AlphaFold notebooks, and you can see an overview of them here on the picture, which are kind of spin-offs of the original idea of getting all of this to run in Jupyter Notebook, but they use more experimental features, mainly this MMStax2, which is an MSA algorithm, which is rather new, at least its application in this deep learning methods is new. It is much faster, but using this is still somewhat experimental. It seems to be doing very well, and there's at least a preprint, but as far as I'm aware, it's still not peer-reviewed, and that allows people to run things in a collab at a relatively good speed. Similarly for RoseTTAFold, also using MMSX2, you can get a speed up and run some version of the algorithm in a simple notebook. What we're trying to do today is actually give you access to what is as close as possible to the actual CAS14 implementations on a cluster, because you could just download this and run it on a laptop, but then you need either a really good machine or a lot of patience. That's why we're trying to show you today is our implementations, mostly Sabri's or exclusively Sabri's work, either on the University of Oslo machine learning nodes, Fox and Saga, which are two clusters here. And this will give you the full experience, basically. So I've chosen a few example proteins, and we'll have to switch to my actual laptop soon, and we'll see how that goes. But these are example proteins I've chosen because we all have experimental structures of them. These are the ones you see. As you can see, there's some stuff close to the proteins that's not actually the proteins. This can be remnants of the solvent used in crystallography, or ligands or cofactors, just to show you that these are the actual experimentally determined structures. I've given you the UniProt ideas. So you can choose one of these three or another one if you prefer one. And then we will try to run these on servers or wherever you have access. So just to go over it, the first one I chose is an enzyme. I like enzymes, and it's not too big and it folds nicely. So if you test this both with RoseTTAFold and AlphaFold, you get a nice result. The other ones are fairly famous, TNF and P53, both play important roles in cancer research. They're a bit bigger, and it will kind of show you where the algorithms are doing well and where they struggle and how they differ from each other, at least quantitatively. And at the end of the last talk, which was very interesting, was this discussion about flexibility and how using this extra information the algorithms give you at the end, you could kind of distill which parts are very rigid and which ones are very flexible. And by that, I mean parts of the protein. And you will see if you run either the TNF or P53 examples that the algorithms do fairly well, or really well in the rigid parts, but then struggle in the more flexible parts. And they also indicate this themselves. So if you want to run Tristan Kroll's very interesting examples on how to split apart these things, TNF and P53 are, especially TNF, I think, would be a nice example. So for the people who want to join, suggest at this point, you go to the UniProt website and you start searching for the FASTA files. I presume most of the people here know how to find them. If not, send a message. I'll try to show it on the laptop, but I'll go through the presentation first because you might have some technical issues with it, as you're noting. I'll come back to the slide. I'm just going to go through the rest first if you haven't seen the ID of the protein we want yet. Okay, the other thing I want to mention is there are a lot of really interesting talks tomorrow, starting from 9 with an explanation about RoseTTAFold and then ending with a very interesting panel discussion at the end. There are 3 talks that are very, very closely related to what I'm doing now. First one will be Sabri and his people talking about how they actually set this up on the Fox server, which is a very nice achievement because I've been able to take the laptop/desktop version of these deep networks for protein folding and get them to run efficiently on a cluster and get as close to the original implementation as possible. The second talk is very closely related to this straight after. It's about what the actual legal side of using all of this, of this implementation results are. And then you've already seen Jon this morning with the very first talk. Yeah, apparently the university clarifies Fox is a server, but not a cluster. I do apologize for the mix up there. So Jon this morning, already gave the introduction, and tomorrow afternoon, or between 11:40 and I think it's something like 2 in the afternoon, he will be talking about predictions that actually come out of our setups, including protein structures that haven't been seen before, so that are not included in AMBL, and what are possible, what possible implications of the structure are. Okay, we will now try to get things up and running. As promised, I'll go back to the slide, I'll give everybody a few minutes to choose a protein they like. So you can either choose this one or look around in the protein database bank, so the PDB, or on UniProt, or just Google what is a nice protein and then type it into one of these databases and get out of FASTA. One thing I want to say is if you go over anything more than a couple of hundred, literally more than 300-400 amino acids, it might take you longer than this workshop to run them, depending on where you run them and which version of the algorithms. So I suggest you take a not too massive protein. Okay.
Jonas, I've given you a new panelist link in chat and email.
Yes, I'm trying to log in.
Yeah.
Thank you very much.
Sabri, in the meantime, someone is asking, what are the specs of Fox cluster?
Yeah, so your presentation tomorrow, it's a very small cluster. I'll give details tomorrow.
Okay, I will stop sharing this. So now you can see me at least. That's something. So I have a bit of issues logging in here on the my laptop. I will try one more time, but it somehow doesn't allow me the option to share a screen for reasons that I'm very unaware of. I've done this before without issue. Even with a new link. Okay, let's try that.
So are you using the link that I sent you in the chat, or you are?
Yeah, it's just sending me to the registration actually for the.
Really?
Yeah. Sabri, can you share a screen with the just a bash file of the commands to run on the cluster? Let's start with that. We don't leave people hanging in the meantime.
So do you want the Slurm script or?
Yeah, Slurm or just a bash for no hoop or something. As long as we have the bash commands for.
while you're solving your technical issues, I can just say that if anyone in the audience now have any questions, please raise your hands and I can open up your microphone. So if you have questions that you want to ask, I can open up your microphone. As all live demos, we do have some challenges, but I'm sure we will be able to solve them. Here is a question, I think, from David.
David Jenkins is raising hand.
Yes. David, please.
I'm not on the live demo, but I have used AlphaFold, and I wondered if there are any, do you have to take particular precautions with the CUDA setup? Because I've been having problems with that.
Are you running this locally, or are you running it on one of the...
On AlphaFold itself.
Yeah, but is it the AlphaFold or the one by Google itself? Because you've got the AlphaFold Colab and the AlphaFold, which are two different.
By now it's one that I've produced, but it was based on AlphaFold. Using MMSeq.
Yeah, I'm not sure about MMSeq. It is faster, so you could just probably try to run without GPUs. But it's fairly experimental. I know they update it weekly, if not daily, so it's an ongoing thing. These AlphaFolds, by the way, allow you to. There was a discussion earlier about this homomer complexes where you have more than one protein and subunits. And the funny thing is that there they allow for this to try and get these more complicated structures up and running. And the interesting thing is that if you say, okay, we have this 6-fold symmetry, you get a nice result as you should. But if you then tell the algorithm, oh no, it's actually an 8-fold symmetry, and that's not what happens in nature, it will also give you an output where you get an 8-fold symmetry for your protein complexes. So people are trying a lot there, and it's hard for us to comment on the technical side of things because we don't set these things up. So I think Sabri has shared your a screen of what you're supposed to run. So whether you run this on a cluster or directly on the machine learning nodes if you have access. So the first part is all just cluster related stuff, which I suppose most of you know how to use. And then the actual part of running it is quite simple. This is the example for first RoseTTAFold. So you purge the system, you load a module in for RoseTTAFold. It's called RoseTTAFold 1.0.0.2 for internal reasons. They're just our current best setup. And then you run this preset run RoseTTAFox bash file with your query FASTA, so whatever FASTA you're interested in, and the dot at the end is fairly crucial. If you don't put it there, the program's not going to run. The dot there is actually the output file, so you're just saying, okay, I want, or the output folder, I want it here, but you could also just give it a name. So running RoseTTAFold is very straightforward. You just call the bash script, put in the query FASTA and the output, and that's it. And then for AlphaFold, it's slightly more difficult, but not difficult at all. It's again running a bash script. Then you have to point towards where all the data is. So the AlphaFold data, these terabytes and terabytes of examples, are downloaded and stored in a place within the cluster environment. So you just have to call on this variable and don't worry about it after. Then you have to again give a folder. Here it's the output folder. And then the M flag tells you which model you want. So AlphaFold has been trained several, or at least released several times. So it's been trained with different seeds, kind of different sequences it's been trained on. And here you just say, okay, I want model 1, and then you get the output from one of the models. And they are to some extent similar, and it will run a bit faster if you choose one of the models. And again, query FASTA. And then you have to give in a time. This time tells you which sequences you take into account. So if you've got a very old protein that you want, well, old by it's been solved a long time ago. And you want to test how the system does on that, you have to move that date to before when the experiment structure was deposited or released. I lost the screen. And then there's some time comparisons, but that's just on our local Fox system. So this is just a current implementation and not a comment on AlphaFold or RoseTTAFold and how fast they run or not. And I'm very grateful to Sabri for kind of helping me out right now. So when you have downloaded one of the uniprods, or it's going to file, so you have this FASTA, so you download the FASTA, and then you write a bash Slurm script. So Slurm is the setup for shared computing resources on most cluster instances, I would say, or at least in academic settings. So I guess most of you are very familiar with this. If not, shout out. But the only thing this does is just say, okay, I have a task that I want to run on the shared computing resource. And this is what I need memory computing wise. Put me in the queue, and then eventually it'll run it.
There's a question, Jonas.
Yes.
For prediction, what is the GPU requirements for AlphaFold 2?
I think we should ask Sabri, to be honest.
Yeah, Sabri.
So the GPUs we use was NVIDIA A100s. So the current setup does not allow us to use multiple GPUs at the same time. So one run is confined to one GPU, but our GPUs have a lot of memory, like four.
We couldn't hear your memory.
Sabri, maybe you can switch off your camera. Maybe your voice will come better.
Yeah, it's for these kind of issues I try to run the Zoom on a different machine than actual computing. I'm briefly going to log out and try to log in just on the laptop that you can see what we're actually trying to do here.
Okay, so we will, if you have any more questions or attendees that want to raise their hand and ask questions, I think we had a question maybe
there was
from...
one from Mario, but I think he lowered his hand.
Exactly.
And there is something from Asmun. Do you have scaling data for number of cores, memory required, and sequence length yet? Maybe this is more for Sabri, but
can you hear me?
Yeah, we can hear you now.
Yeah,
Sabri.
Sorry, I'm on a mobile internet, so due to some internet issues. So the thing is, AlphaFold and also RoseTTAFold, they will run only on a single GPU at the moment. We have not done this shared memory installation, but they need a lot of memory. So the processing done here that we shared on GitHub is using a 40 gigabyte NVIDIA A100.
You're breaking again, Sabri.
I suggest, Sabri, that you can answer in the chat or at the Q&A. We do have some more. Yeah, so Sabri, please enter the GPU card that you are using. So please do write this
I think
in the chat.
It was A800.
Okay,
Jonas.
I still don't have access on the machine, but I'll keep you company until I figure this out. So if somebody who can actually share a screen so far could.
Yeah, if Sabri again could try to share his screen and show.
I do apologize to everybody who's joining the webinar. Tested the setup.
This is exactly what happened. It's supposed to happen at the live.
Yes, this works perfectly when you test it. And, of all the things I expected, not being able to share the screen was not the one.
But Jonas, I don't see your another account right
now.
No,
I've locked out with that because I just didn't get access on.
Because we can try and make you co-host if that gives you a better.
Yeah, but even with my normal account, it doesn't like to share. But if you give me a moment, I will try to figure it out. So for the people who have access to the clusters, you can run what Sabri showed you.
We can also try it this way. I don't know if you have a Google Meet that works on browser, and you can just share the Google Meet link here, and we can all move there. I mean, we have no love for Zoom, so it's only the hands-on session right now. We don't have any program after this. So if you want to try your browser with a Google Meet, that should be fine.
I will first test it, and then if that works, then we can try that.
I will propose that we will take a short break, maybe 10 or 15 minutes. And of course, anyone are welcome to stay here, but then we can try to sort out some technical issues. So if we take a break until 2:35 Norwegian time, and then we will see if we can come back with some more hands-on. Does that sound like a plan, Jonas?
That sounds like a plan.
Yeah, so a bit more than a 10 minutes break now. Please pick up a coffee and we will see if we can solve the issues. Okay, thank you. Okay, Jonas. Of course, everyone is still here, but we have a break. Do you think you will be able to share a screen from?
Somehow it's not allowing me to select the screen to be shared. Okay. And I have no idea why, because I've literally done this yesterday without any issues. I don't know if it's an updated browser or whatever. I'm just gonna close.
Because you have multiple screens, right?
Yeah, but one is on the iPad, so that all runs okay. But I'm trying just sharing the screen on my Firefox on my Ubuntu is for some kind of reason not working, even though it was like yesterday. I'm just trying to think if there's another way to do this. It's not.
Can we try this, Jonas? I've given you a Google Meet link. If it works on your Ubuntu, I can try to share this screen from my side on the Zoom. So.
Oh, I know what's going on. Yes, I know what's going on. Okay, perfect. I'm going to try and I'm going to log out. I'm going to be back. I'm going to solve this.
I can hear some optimism. See you soon. Perfect.
I know what's going on. It's I've got an external screen on my laptop, which is interfering with the whole thing. My apologies for this.
No problem. I will go and pick up a coffee and we'll be back.
Yes, and then we will restart this thing with.
Yes, perfect.
10 minutes. 10 minutes.
Yes,
10 minutes.
I will be with you.
2:35 Norwegian and European time, Central time.
Yes.